Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. My guest today is Mahmoud Ghannoum. He's a professor in the Department of Dermatology, also part of the School of Medicine, part of the Department of Pathology as well. And we're going to be talking about the microbiome and the mycobiome, which is fungal-related microbes in regards to, I believe, cancer. So, Mahmoud, thanks for coming.
Thanks for having me. It's really great to be with you.
Yeah, if you would tell me about your research, what's the focus of it today?
My research these days are really focused on as you mentioned, the microbiome, which is really the microbial community or the community of organisms, bacteria, fungi, viruses that live on our body, like the skin, for example, and inside us, such as the gut. And we are trying to look at how these communities can affect our health and wellness. Clearly, I'm focusing at the moment in a couple of areas. One is the digestive health and the other cancer.
So what types of cancer are you looking into?
We are in particularly looking at two types of cancer: tongue cancer as well as head and neck squamous cell carcinoma.
Mm. And have you looked to see, I know there's a microbiome of the mouth, but of the esophagus, is there a different one? Like, have you observed that microbiomes are everywhere, or are they confined to certain areas in the body?
They really, they are everywhere. They are inside us, as we said, is the oral cavity, in the gut, in the reproductive system, but also they are present in our skin, in our nose, our ears. And we even published work on the eyes, like contact lenses, as well as the microbiome on the eyes. We have some microorganisms, especially if people use contact lenses, you may have more microorganisms. In particular, we looked at a fusarium, which is a fungal infections, which is present in people who used to use some lens cleaning solutions. So you can find it all over our body.
Yeah, amazing. In regards to the cancers you're studying, do they form tumors? And if so, is there a different localized microbiome on the tumors versus the regular tissue?
That's exactly actually what we did. We looked at, in collaboration with two very good friends and colleagues. One is Carice Eng from the Cleveland Clinic, and the other is Dr. Ted Teknos from the Seidman Cancer Center at University Hospital. We are working in cancer in general with these two friends and colleagues. But with Carice, what we looked at the tongue cancer, as well as I mentioned, head and neck squamous cell carcinoma. So let me tell you what we did with the tongue cancer. And before we go in detail into what we did, the idea came from the fact that several studies, epidemiological studies, tried to look at the relationship between poor oral hygiene, in other words, the people don't brush their teeth, they don't take care of them, and cancer. And what they found that really there is a relation. And what helped us a lot in these studies is that there is a new sequencing technology which allows you to identify what organisms there very fast, and also allow you to identify both those that are easy to grow and those that are difficult to grow. And what people found is that overall, it looks like if somebody have an imbalance, or they call it, the scientific word is dysbiosis. In other words, the microbes living in our mouth are not in harmony together. They are out of balance. And they found that if we have this imbalance, it is likely it will contribute to cancer or tumor genesis of the mouth, not as well as other parts of the body, such as the gut, the lung, and so on and so forth. Okay. And that's how it started. And then we came into the picture because a lot of the published work looked only on bacteria, or called the bacterial community. And our work looked, in addition to the bacteria, we also looked at the fungal community. People thought there is only bacteria in our mouth. And I can tell you, based on studies which I did many, many years ago, I knew that both bacteria and fungus exist together. And how did we know that? If somebody, a lady, for example, take an antibiotics, you're killing the bacteria, and guess what happened? They develop thrush, and thrush is caused by fungus. And that what taught me that if we affect the bacteria, we give a chance to the fungus to start causing issues, okay? And that's why we thought, okay, people looked at the bacteria in cancer. Is there any relation between fungi as well in these patients? So we went and enrolled patients, about more than 50 unrelated patients, where they had mobile tongue cancer, the type of cancer they had. And we collected tissues from them, from the cancer itself, okay, and adjacent normal tissues. So we tried to compare both what is the microbiome, both bacteria and fungus, in the tumor tissue in the tongue and those that are not, okay? And so with that, we looked at different points there. We tried to see what is the diversity. To make it simple, diversity, it means what type of organisms are there, how many different organisms, both bacteria and fungi, and also at what levels they are. Like, sometimes we call it abundance, like the level, like is it present 10%, 15% relative to the others? So this is the level. So what organisms are there and how much of them is there, okay? And also, that's what we looked at, both bacteria and fungi. And to our surprise, we found that both really bacteria and fungi are affected in cancer tissue versus non-cancer tissue, okay?
How would you say they're affected? Like, is there a totally different regime for the microbiome, on a tumor, or is it only slightly different?
Yes. So what we found is, as I mentioned, the diversity, like how many organisms are present. We found that the diversity decreased in tumor group, like the patients, the parts of the tumor, when we looked at it, we had less number of organisms, okay? That's in the bacteria. Whereas in the fungi, we found that the richness, which means how many actual species are there, it was really decreased in the fungus. So we had less fungus in people with cancer, okay? Then we start to look at, like as you alluded to, what sort of organisms are there? Are they different species? Do we have different bacteria? Do we have different fungus? Okay. And then what we found, in cancer patients, some organisms increased. Like, for example, one group of bacteria called the Streptococcus, we found that it increased in tumor group, and whereas other organisms, we found them to go down. So you can see the balance is shifting, and that's why they become imbalanced. And the ones that really decreased in the tumor group, like example, one very well known called the Fusobacterium, okay? So you can see some organisms increase, some organisms decrease, okay? And then when we looked at the fungus, we also found a large number of them decreased, okay? Such as Aspergillus. This Aspergillus is a mold which is present all over the place, but you can find it in people, and it could cause disease as well. So really, basically, doing this study, what it told us is that there is a difference in the bacteriome, which is the bacterial community, as well as the microbiome, which is the fungal community, between the tongue of cancer patients and their matched normal healthy people. So you can see there is a shift in there, you know.
Did you do
And
any transcriptomics or metabolomics on it to see, what's the consequence of that? This different regime, what's it producing?
you honestly, you took the word out of my mouth, because
Mm,
we did not look in tongue cancer about the metabolomics, but we looked at metabolomics in the head and neck squamous cell carcinoma. Okay, so the first study we just looked at the microbiome, but in the head and neck squamous cell carcinoma, we looked at what you call metabolites. Metabolites are chemicals that we can characterize very easily using metabolomics. This is new. Again, we are lucky because the techniques of capturing these and identifying them now, it's like with sequencing, is really very advanced and allow us to look at this. And in this group of patients with head and neck squamous cell carcinoma, we really tried to took samples similar to what we did with the tongue cancer, and we compared the chemicals that are in cancer versus non-cancer. Okay, and
okay.
to our amazement, we found that there were 22 different compounds or metabolites we were able to detect, and eight of these 22 were statistically overproduced in the cancer, which means there's more amount of them. The level of the chemicals, if you look at how much there, it was increased in cancer tissues. We dug a little bit more deeply, and we identified one particular chemical called 2-hydroxyglutarate, and we found that it's present only in tumor tissue, but not in healthy one. Okay? And this really is very interesting, because this suggested to us that it is possible to use this chemical, 2-hydroxyglutarate, as a biomarker of disease. It looks like if you find it, it may tell you that this person have cancer. And the beauty of this is that it is a non-invasive process. Like, you don't have to take biopsies, which is, as you know, very painful sometimes to patients, but we are able to do it with the oral wash, for example. Or sometimes you can do swabbing of the mouth, and you look for these chemicals. But in our case, we used oral wash. And now what we are trying to do is we would like to conduct some clinical trials to really validate this observation and see, will it stand? Like, we would like to take some patient and do the traditional way of diagnosing this cancer, and see whether we can see this compound and how it's what you call specificity and really sensitivity. You need it to be
what
very.
creatures produce it?
Yeah, we did not find out which organisms produce it in this case, but that's another thing which we hope to put an NIH grant to try to identify these metabolites.
Is it hard to figure out who's producing what substances?
It's the nice thing I can tell you. I just got funded from the National Institute of Health to look at metabolites in Crohn's disease. Okay. And what we did, we identified organisms like we did in this cancer patient that are increased, elevated in Crohn's disease patients. And what we did is we took these bugs or microbes and we grew them, and then we did metabolomic analysis. And we identified a few compounds that are secreted by these organisms. So now the grant which I got from the National Institute of Health is to do what? Is to see what can these chemicals do to cause Crohn's. So we will use this similar approach for the cancer, but we haven't started that yet.
So how do fungi seem to act in a normal microbiome versus a dysbiotic one? You said sometimes they appear to take over. I mean, what other interesting behaviors have you seen?
What's so interesting about the fungus, if I can tell you, I did a study many, many years ago to see how many people have Candida. Candida is one of the fungus that cause a lot of trouble.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
Okay, it causes thrush in the mouth, in the vagina, and also it causes systemic diseases in cancer patients. And you will find that based on our study, 50% of people can have this bug, this fungus.
Wow.
But it is at low level, which means it really does not cause any issue if your immunity is good, if we have good bacteria that keep it under control. And the same applies for the gut. In the gut, many of us have it. And what happens when we take an antibiotic, we are killing not only the bad bacteria, we are killing the good bacteria as well, which keeps Candida under control. And when that happens, guess what? Candida start to overgrow and cause issues. So that's why now you may ask, you telling me that they are present at low levels. Are they helping us? I can tell you at low levels, yes, they are helping us. Like, for example, there is a study done where they looked at people with low level of Candida, and they found that it cooperates with the bacteria in breaking down food, fermenting food. It breaks the food, which gives byproducts to the bacteria to eat, and then bacteria can help them back. So it's really a very nice beneficial cycle. So that's why understanding how these organisms work together is very, very important.
Yeah, what kinds of fungi are in someone's typical microbiome in their tongue and their throat?
You can, what we found in our study, the first study which I published is in just characterizing the microbiome or the fungal community in healthy people. I just wanted to see what is there. And you can find Candida, you can find Saccharomyces. Saccharomyces is a good yeast, it's the one we use in baking bread, in making beer, for example. You can find other organisms such as Cryptococcus. Cryptococcus is one organism which can, in HIV-infected patients, it can cause issues. But you can find it normally there. And that's why I think when it is normally there and somebody's immunity go down, then it starts to cause trouble. We found also Aspergillus there. So you find about 15 different organisms are present in the oral cavity, and they are present in more than 20% of the people, because sometimes you may have fungus that is transient. You eat, let's say, taco, and there is some organisms that live there, you will see it. But what helped us is we looked at large number of people, and we looked at what are the most dominant fungi present in at least 20% of the people. Otherwise, you can consider them transit. And
right.
Based on this analysis, we found about 15 different fungi. And in a person, we found between 7 to 9 different species.
Can you tell if the bacteria interact with the fungi in various ways? Like, could you experimentally look to see what's being passed back and forth between them, if anything?
Sure. We did. We published actually a study in 2016. What we showed is bacteria and fungus, they come together. Especially, let's talk about the bad ones. We can talk about the good ones. I just mentioned some to you with respect to breaking down food. But when it becomes in a pathogenic state or virulent state where it can cause disease, what we showed is that these pathogen bacteria, and two types of bacteria, E. coli and Serratia marcescens, as well as Candida, and one species in particular called Candida tropicalis, they come together and they form what we call biofilm. What is biofilm? The simplest way to explain it, it's like the plaque in our teeth. In the morning, we brush our teeth. In the evening, we brush our teeth to get rid of the plaque, because these organisms, they come together and they form like jello. If you can imagine, you have a jello, and inside this jello, we have raisins or M&Ms. The microbes are raisins, the M&Ms. Whereas the jello, it's what these organisms secrete. It's like viscous material to keep them inside the protected. So we looked at that and we were able to publish that work in Crohn's disease patients. And I had a lot of experience in this biofilms before I went into the microbiome. I used to study these biofilms, like for example, if somebody goes to hospital, they put a catheter in them, to give them drugs, to give them their food, if they can't swallow. And guess what happened? These organisms, they come, they stick to the catheter, and they start forming biofilms. And when you have these biofilms, you can't get rid of them, and you have to take the catheter out. And they cause what you call catheter-related infections. That's why I was lucky when I start looking at the microbiome. I also saw that this interaction where bacteria and fungi come together and form this biofilms, which could be detrimental to us.
When you've cultured bacteria, from the nose, the throat, from the tongue, are they going to be typically free living, or are they going to be in biofilm format on all those surfaces? And what does the structure of the biofilm look like, if so? Okay,
I think in the gut, in the mouth, in the vagina, they form biofilms. We used to think they live like in suspension, but no. What they do, they stick into the material, what we call they adhere to our mucosal tissue, whether it's the nose, the mouth, or the gut, or the vagina. And once they adhere there, they start to produce what you call polysaccharides, like carbohydrates, sugars. And these carbohydrates is what forms the jello. It's like a protective layer. They are inside it. And that's where it becomes very difficult to try to kill these organisms, because they become resistant, not only to antibiotics and antifungal, but also they become resistant to our immune cells. And we published that work. We showed how really we took some immune blood cells, we made biofilms, and we tried to see whether like macrophages or neutrophils are able to go and attack these pathogens, and they really did not. So having a biofilm, it's very difficult to treat, because as I mentioned, a hallmark of a biofilm is resistance. So we need to break it down.
Are you looking for therapies to these types of cancers in terms of influencing the microbiome? Like do you think that if you were able to nudge someone's microbiome from a dysbiotic state back to a healthier state, do you think that would affect their cancer significantly?
Definitely, this is one way to start looking into how can we prevent cancer and how can we help, And definitely we are doing, that's why I co-founded a company called Biohm Health. And what we started to do is develop probiotics that can rebalance and maintain the balance of the microbiome. The first product we had really was based on our Crohn's disease studies. And now we are starting to look into the cancer, what sort of things. In fact, we have a clinical trial to start very soon using this probiotic we developed in breast cancer to see whether it can change the microbiome or not. But this is just going to start in the very near future with Dr. Eng.
What about phages? I know bacteria have phages, especially in our microbiome, but what about the fungi? What do you call a fungal phage or a fungal virus? Just a
we don't have those. Like in the bacteria, we have the phages, which, as you say, kill the bacteria, but not the fungi. We don't have that. It's really, the fungi is similar to us. It's the eukaryotic cell. It has a nucleus, unlike the bacteria and this sort of thing. What we try to do is we try to interfere with its virulence factors. Like, for example, what can we do to inhibit its ability to invade tissue? Let me give you an example. If you take Candida, it is like baker's yeast. It is in a shape, elliptical, small round circle. When it is in our body and it wants to invade the tissue or break down the lining of the gut, for example, it starts to form what we call hyphae or filaments. These filaments, they start, it's like you have a needle start to go through our gut lining and invade the tissues. So what we want to try to do, how can we find new ways to prevent candida from forming these hyphae? So that's how we are trying to attack that. How can we ensure that candida does not make a biofilm? Because in our study, when we looked at the biofilm, if you put Candida tropicalis alone, it was all like little oval circles, oval shape bodies. It's like yeast, like Saccharomyces cerevisiae. Okay, it's like yeast, the same shape. When we add the bacteria to it, these two organisms, E. coli and Serratia marcescens, guess what happened? It was able to start forming what you called hyphae. And this hyphae have been shown to be a virulence factor. It's one of the properties that Candida use to cause infection and break tissues. So now we are trying to develop a probiotic that can inhibit the biofilm. And in fact, I published a paper in a journal called MBio. It's the American Society for Microbiology, where we showed that the three probiotic strains that Biohm developed can break down this biofilm. So this is our approach now.
So if you prevent the hyphae from forming with Candida, you're preventing these biofilms from growing, and then it's easier to attack the fungi to get rid of it.
Exactly, absolutely. This is the right way. And also it can be eliminated then, whether if it's in our gut, it can be eliminated, by going to the bathroom.
Well, is Candida, is it there in low levels in healthy people, or is it not there at all?
No, it is, as I mentioned, it's about maybe 50% of the people have Candida when you are healthy. But it's no problem, because the only time it starts causing trouble is when you kill the bacteria, which keep it under control, or if your immune system become weak. Like, for example, cancer patients, we give them so many drugs, chemotherapy, to kill the cancer cells. And what happens, it weakens our immunity. And because our immunity is weak, Candida then, or fungus in general, to be honest, start causing infection. And that's why we call them opportunistic infection. They take the opportunity when our system is weak, you are under trouble. However, if your immunity is good, you are going to be fine.
You don't think that there doesn't appear to be any phage or viruses that affect, the fungi?
Not really, not in this sort of situation. But the phages, as you know, people are using it to control bacterial pathogens.
Yeah, I just wonder again if fungi can produce phages, but I didn't know if they could or not.
I don't think
fungi
so.
have phages. Yeah.
I don't think so. At least so far, we haven't seen.
So, what method did you figure out to stop the Candida from making hyphae? Like, what was it treated with?
We treated it with the probiotic which Biohm developed. We have 4 different strains, as well as we have an enzyme amylase, which have been shown to break down biofilms. So when we put them together, they were able to break the biofilm. And as I mentioned, we showed it in vitro, but also we showed it in an animal model where we made biofilms. We put the organism, which is Candida, E. coli, and Serratia. They were able to form biofilm. And then when we gave it the probiotics, the biofilm did not form. Basically, it broke it down.
So that means certain bacteria are inhibiting the creation of the fungal biofilm or the hyphae.
Yes,
right?
Yes, exactly. The beneficial bacteria.
Huh. Do you know what kind of trading goes on between the fungi and the bacteria? In the mouth or other places?
Yeah, it's like what we mentioned about they exchange. For example, we found when you put the 3 organisms together, they start to produce these metabolites. And one of the metabolites which we found in the case of organisms that cause Crohn's disease, it's called 5-oxyproline. And this 5-oxyproline really is increased 130-fold higher than if you take Serratia which is the bacteria by itself. When you put them together, as if they start talking together, and then they start producing this chemical, which allow Candida to start forming the hyphae and, of course, invade. Yeah, we are correct. That's what is the NIH grant, which I am doing now, trying to understand the mechanisms by which this interaction between bacteria and fungi in Crohn's disease.
Oh, very interesting.
Yeah.
So what's next for the experimentation? What needs to be figured out from here?
This is really a very good question. What we are doing now, Biohm Health, we have a gut testing kit where people send us their samples, and we are collecting data under, of course, their consent to look at the microbiome in different conditions. Like we have people who are obese, people who are, for example, autistic kids. We are looking, and now we are trying to characterize what is the dysbiosis. Once we know the dysbiosis, we are hoping to come out with different approaches, probiotic-based, as well as in certain cases, especially disease, you really need to go after biotherapeutics, because this is like a drug. You cannot claim using probiotic that you can treat a disease. Like in the case of autism, we did a very nice study. We identified what organisms are there, and now we are designing a set of biotherapeutic strains that hopefully it can bring the balance back and maintain it and help these kids. So this is where we are going with, is trying to use our big data that Biohm Health developed to come out with new products to address different diseases and different conditions.
Okay, well, very good. So
Thank
Mahmoud, what's the best way for people to find out more about your work? Where can they go?
They can go into biomhealth.com. We are putting a lot of information there. Also, there is the Microbiome Report, is a podcast by Andrea Nguyen. It's she's a nutritionist. It provides guidance with respect to the microbiome and all gut issues there, and hopefully that will help them.
Okay, well, very good. Well, thank you for coming on the podcast.
It's my pleasure. Thank you for having me again.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.